Kia ora everyone, ko Andrew Veale, toko ingoa. Something that I've heard is that eventually you start to resemble your supervisor. So hopefully, I mean, we both look very young, and so yes, Diane was my PhD supervisor on stoats, which I have maintained, and probably I'm going to stay on stoats forever. And so I actually did get a tattoo of my stoats genome. So particularly I'm going to be talking about stoats today, and yes, I worked on the stoats genome, and this is one of the chromosome level ones. You can see the telomeres on the ends. And that's because we threw vast amounts of money at it, and with long read sequencing, if you have enough depth, and every sequencing technology basically, so we had high coverage PacBio, 10x, BioNano, Hi-C, and everything's there. 99% of it is in chromosomes. Feel free to have a look, and we have been using that for various things in terms of trying to work out how to control them. We've been doing quite a lot of GBS, genotyping by sequencing. Basically, you chop up the genome, you sequence the bit next to the cut site, and it's the same 1% or so of the genome. Then you look at those bits and you find the variable sites. So that gives you a lot of markers. And so during my PhD, I used 17 microsatellites. Now, with the studies I'm about to show you, 30,000 SNPs on Waiheke Island and 56-ish thousand SNPs in Taranaki. And that gives you a lot more resolution to do things with it. Also genetically determined sex, and that's a very useful thing to do, particularly for degraded samples. Now, I'm going to talk about Waiheke Island. Currently, they're trying to eradicate stoats on Waiheke Island. I would say that this is about the only true eradication project that's been funded by Predator Free, and hopefully they will remove stoats from that island soon. How are they doing that? By trapping. That's the trapping grid that they have. So that's approximately a 250 by 250 meter true grid, which means that there is at least one trap in every stoat's home range. They have now got, I can't remember, it was 100,000 trap checks. They're doing it now, many of these traps, more than weekly. And so they've been doing it for 3 years. I have been getting all the samples off them for the last 4 to 5 years. And so I have sequenced every stoat from that island for that entire time. Now, the genetic diversity on Waiheke Island is low. This is looking at the inbreeding coefficient from the Auckland region on the mainland, the 2 stoats that were caught on Rangitoto beforehand, and then Taranaki and Waiheke. So the most inbred stoats caught on the Auckland mainland are about average for Waiheke Island. So that's sort of potentially full SIB mating. I think that quite possibly the ones on Waiheke Island were founded by a pregnant female. They're very different to the Auckland ones, and there is no ongoing immigration. So if you look at the genetic distance between each stoat and each other, and the geographic distance between them, ideally you should have a nice one where the ones caught close together are more related, and as you get further away they're less related. You do get a little bit of that pattern. And so I've just taken the ones in this box, which are likely to be first order relatives, so full SIBs or parent offspring pairs, and looked at the distance from them, and you see this interesting pattern. You get ones that are very closely caught together, often in the same trap, and these are ones that are in the same litter, or mum went into the trap and then the kids went in. And then you get this interesting secondary peak at about 3.5K, and my interpretation of that is that's the average dispersal distance where they run along and then they go, okay, I'm probably out of my parents' home range. Is this a nice place to set up shop? And then they go into a trap. But you are getting ones on this island that are first order relatives that are 12 kilometers apart. This is an island that is 20 kilometers long. So stoats can just run laps on Waiheke Island, which is why you don't get much signal in terms of looking at them close to far away. They can disperse much further than that. The furthest ever known dispersal of a stoat was 65 kilometres, and that was, I think, in about 3 weeks, and it was ear tagged in one place, and then 65 kilometres away it was caught. So stoats are really difficult. Looking at the relatedness of all of the stoats, this is a heat map, so it's all versus all. The redder it is, the more closely related, and more yellow, they're less closely related. Now, if I was to do this with all the stoats in New Zealand, include Waiheke Island, they would all be dark orange. They are all very similar, but with 30,000 markers, we can start to get this. Now you get these interesting square structures. These are often either litters if they're small, or multiple litters of closely related individuals. And what I've done on the side here is marked ones that were caught since September 2020. And you get that, so there's 2 older males. I've aged every single one with measurements of their baculums and their skulls. So this is the most studied invasive population ever, where basically every single one has the exact age, location, and genomics. And I got between 3 and 5 litters. There's a little bit of uncertainty on that just because some of them were related individuals, and I think that actually there was also a litter where most of them weren't caught the first year and they were caught second year. So I think it's probably about 4 litters, but anyway, we're getting that level of pedigree analysis done. There's also not actually great software for analyzing full pedigrees of genomic data, so I've been using various Python scripts to get into various things and trying to get the level of coverage right, because you obviously lose a lot of markers. If anyone has any great ideas, but I have got largely the pedigrees worked out. Most of the time they want you to have the exact age. I'm getting that, but often it's hard. So this is the sort of information we get. Now there were 2 litters that shared one individual. The earlier red ones were the one, and then the orange one is the one that shared between them. She was the daughter in the first litter. Then, so she was caught in November in 2020. A couple of days later, same trap, caught one of her sons. And then the other ones, the other yellow ones, are her offspring. And that's 7.3 kilometers. And so one of her kits, who was, without a parent in quite an early age, it dispersed across the island despite all of those traps 7.3 kilometers before it was caught. That's really scary because it obviously encountered traps and avoided them for months as a juvenile. This is more recent stuff. These are ones that were caught this year. Those red ones are the litter. Dad is the yellow one. Mum was never caught. I know this because I have the age of every single one. And when you're doing every single animal that's caught, you know that she's missing. So I said, bring the dogs in, probably because that's the earliest one, you need to find her there. So they did, and there's a den, exactly the point that I pointed out on the map. So they're currently trying to catch this one stoat, which is indicated through a lack of a, mother in a pedigree from Genomics. Great win! Also, the one above is probably an offspring of hers, and they have actually live trapped it, and we've brought it to Landcare to do work on it because it's one that avoided traps forever, and they surrounded it by about 50 traps because they had footage of it going in and out. Eventually they live trapped it. It's a very unusual stoat in terms of behavior. I have seen it. It's very ninja, doesn't want to be anywhere near people. So we're trying to work out, well, what is the behavior of a stoat that avoids traps? Because that's the one that we want to know. To my knowledge, this is the first ever eradication using genomics and full pedigrees to try to get every single animal. And between 3 and 5 litters were born, and some females on the island were caught that did not have litters, as far as I can tell. That's amazing because stoats, all females are pregnant. They're impregnated before they open their eyes, and that means that there were ones that never encountered a male in that area, so they're getting them. They're very close. Also, I've shown that multiple times that relatives have gone into a trap shortly after the other one, which means that scent is important, so they're now rubbing bodies all over traps. And they're looking for dens right now, and they recently caught a lactating female, and they're very close, you know. But the thing is that this has been going on for many years, and having 3 to 5 litters every year, that's a control operation. It's a very good control operation, but it's not eradication. So it may be impossible to actually eradicate stoats with traps, and if they can't do it because there's zero immigration, no one can. So moving on, in Taranaki, we've been trying to look at this with radio collars and GPSs, trying to work out how they move across this environment, and they have huge numbers of traps. I can't remember, it's tens of thousands. And unfortunately, all my samples have disappeared. Great. There used to be something else on that figure. But I identified 77 close kin we got 600 samples, 77 close kin for stoats and only 2 for weasels. That wasn't great in terms of being able to work anything out with the weasels. And I have sexed them all using genomics. And so this is the read depth ratio on the X chromosome versus the autosomes and also the Y chromosome. Definitely the best way to sex them is looking at read depth on the X chromosome, because even in samples that are highly degraded, there's going to be some reads on the X and some reads on the autosomes. So we can sex every single one. Sex ratio for stoats was about 1:1. Great. That means that they aren't missing any because of the stoats' female home ranges being small and the trap's not getting them. Weasels, it's 3:1, so they're missing a lot of female weasels, and they knew that this was probably going to happen, and that's because DOC 200 is set to about 100 grams, female weasels about 50 grams. They're making weasel paradise because all the female weasels aren't being caught. So that was important. Now, just looking at the stoats, this is a PCA so PC1 versus PC2 of the genomics, and you can see there's a little bit of a pattern with PC1 being stretched and then PC2. Now, if you look, so I colored the map by PC1, and it's just a spectrum. It's just isolation by distance-ish. But, and then PC2, that funny group, that's all these ones caught down near Hawea, and it's just that they were far away. That's all it means. So there's no big population structure, there's no barriers to dispersal. And when we look at close kin, that's what they're doing. There are ones dispersing 40 kilometers. There's 2 40-kilometer dispersals there. They're probably males because it was one male and multiple other ones at the other end. So, and that's within an environment with 40,000 traps or whatever it is. It's a huge trapping regime. They are not catching them, they're dispersing that far, and so basically if we were to use magic and there's no stoats left in Taranaki, how thick would you have to have it be to prevent them not reinvading? Probably 60 kilometers of 250 by 250 meter grid of traps might stop most stoats. So yeah, these are the 2 dispersals for weasels, and weasels are tiny little guys, and they're still, that's like 20 kilometers one dispersed. So I made a nice dispersal curve, and it's not informed by any landscape features because they're just moving across everything. With possums, it's much better. You do get these kin groups, is what I'm calling them. I'm trying to figure out how to do it better than this, but you get these nice colors. Also, look, all the ones that are caught in the national park are the same. In the ring plain, there's lots of little groups because there's less connection, because they don't like going across farmland. Really cool stuff. If we look at rivers, they're barriers, but not quite. They definitely seem to cross at bridges and at the beach, but you do get these little structures, and this is just looking at family groups. We're not able, this isn't population structure, this is kinship structure, and it's something that I'm trying to figure out how to work out how to do. And then we are creating these maps of where they'll come, okay. And this is just a difficulty with that, is that you can't actually, so this is looking at relatedness, trying to find a measure that is actually able to inform these maps. So anyway, basically I just talked about the landscape stuff and the pedigrees, but we are doing lots of other things potentially to inform stuff. So I'm happy to answer questions on any of that as well. Great.
quick questions?
And
It's awesome, Andrew. Well done. Is this on? Yeah. And it is just like looking in the mirror. The Waiheke Island stuff, that's amazing, because I remember, I mean, obviously that was, you know, part of your PhD at the time, but are they, like, at the time in your PhD, when you did that first stuff, we assumed that they were swimming there. Are they swimming? They are swimming there?
I believe that they did swim there originally, but there have been occasional stoats on Pōnui, so they may have swum via Pōnui. It's a 5.2 gap, and I think that occasionally they can do that. Rangitoto Motutapu has now had 6 reincursions, all independent. That's 3K.
Yeah, okay, yeah.
But with the microsatellites from my PhD, they were all basically clones, and I could get the stuff back and just eyeball it, and oh, that's a Waiheke one.
Yeah.
If you have 30,000 markers, now you can get a lot more,
yeah.
And you can start getting that kinship stuff. The reason that there's only 30,000 and 60,000 in Taranaki, they are so inbred, everything's fixed.
Wow.
I honestly believe it could be a pregnant female that founded it.
Wow, that's amazing. That's really good work, Andrew.